Ti ho fatto la mail con il nuovo link sotto.
Sì, dai Julio. Io ho sempre voluto vedere la mia foto,
Buonasera a tutti.
Buonasera. Hola.
Bene.
Okay, Julio. If you want, you can start. Thank you.
Okay. There seems to be a delay with the sound.
Yes. Yes.
But did you hear me okay?
Yes.
Okay. let's try. So this is funny because I'm hearing myself. So you can see and hear me okay, correct?
Yes. Yes.
All right.
Okay,
Go ahead.
let's go. So, well, thank you for having us. Yes, so what I'm going to show you is some work that some of the postdocs that work with us here at CESE have been carrying out in the last couple of years. And it has to do with the coupled physical biochemical modeling of the Gulf of Mexico. So particularly, I'm going to show you the work of Pierre Damien and Sheila Estrada. Before I do that, let me talk a little bit about what we are trying to model in the Gulf of Mexico regarding the biochemical variability in the region. And so probably you, I don't know, you may see this already during the thing. This is the chlorophyll concentration at the surface, the climatology, remotely climatology of the chlorophyll concentration at the surface. You see on the left from November, December, January, April, and then May, etc. So on the left-hand side, you have essentially winter, and the right-hand side, you have essentially summer. And you can very clearly see that there is a seasonal cycle in the chlorophyll surface concentration. But you can also see, I hope it will work, I'm not sure. I hope you can see this. I'm using my mouse. Do you see it?
Yes. Yes.
So notice a couple of things. And you can see here the outflow of the Mississippi River already here, and then these jets here at the border with the US and Mexico, and another jet around here, which is supposed to be related with the convergence of the coastal currents in this region, but also with the interaction between the coastal currents and the eddies, particularly because you can see that, that these jets actually go all the way to the middle of Mexico. So it's not only the coastal currents, it's really the contribution of the eddies in the region that are producing this kind of patterns. The other interesting area, again, is the upwelling in this region, which is the Yucatan Peninsula. And you know, in this, the Gulf of Mexico is dominated by trade. So these coastal areas are prone for the standard upwelling mechanism by the wind. But actually, what happened is that upwelling in Yucatan is really controlled by the Yucatan current and the loop current. I will try to show you that later on. Besides this, there is the network of observations, one from the SIGOM project. This is the left-hand side, you see the map of CPD and other biochemical measurements that have been carried out since 2015. And this is really filling a gap that we have for many years, in which we didn't have any really any observations on the Mexican side regarding many variables. And on the right-hand side, you see the patterns of BOEM. This is the Bureau of Ocean Energy Management from the US that funded a project in which 6 bioargo floats were deployed in the Gulf of Mexico for 5 years. And that also provided an unprecedented data set to analyze what was going not only at the surface, but also at depth. So into what we are trying to explain with the models, these are results from a paper and work that French postdoc that worked here at CICESE 2016, Orange de Fourmevaux, Pasquerol de Fourmevaux, and with all our group, he worked on analyzing this bioargo flow data set, and many interesting things came up from this study. The first one was, if you look at the figure of in the bottom left, these are time series from well, many of the, or several of the bioargo floats. In white, you see the mixed layer depth. In color you see the chlorophyll concentration, and it's very difficult to see, but there are some gray lines which are supposed to be the isopycnals. And what Orange noted was that a clear deep chlorophyll maximum during the year, but also during winter, is that these areas, let me see, this increasing of the chlorophyll all the way to the surface was really apparently produced, or essentially produced, by mixing processes. But then what happened is, now you have to look to the right-hand side, where you see the mixed layer depth, the surface chlorophyll concentration, and the critically integrated chlorophyll concentration. And what he found was that vertically integrated chlorophyll content was relatively constant during the year in all these aqua flows. This was kind of surprising, really, because the idea was that this seasonal cycle at the surface, where you see the increased surface chlorophyll concentration during winter, was, or at that time, was thought to be produced by new production, if you like, an increase in biomass really at in the euphotic layer. What essentially we thought is, well, this is not really happening at the basic scale. This is something important to mention, because even though we had like 700 argo profiles, the profile, each profile or each argo float measured or make a vertical profile every 15 days. And so, the sampling in time is not very good. But anyway, the idea is, well, we found this thing that the integrated chlorophyll content was relatively constant, and essentially, if you look to the left, with what we came up as an explanation, was that what was happening is that really no new nutrients were not really getting into the euphotic zone during winter. What was happening is that the mixing during winter produced an homogenization of the peak chlorophyll maximum, but it didn't really, introduce new nutrients into the orthotic layer. And so we actually, in this paper, we also did a separation between cyclones and anticyclones. But basically, the idea is that in some sense, even though you have a little bit more chlorophyll concentration, integrated or total chlorophyll concentration is higher in cyclones than in anticyclones. What was happening is that the nutricline was really slightly deeper than the deep chlorophyll maximum, and the mixed layer was not reaching this nutricline, and that's why we didn't get any new bio, an increase in biomass. So these are the kind of things that we are now trying to reproduce with the models, and use the models also, if we are able to do that, to also look at other properties that we can actually compute directly within the model. So the first model, modeling results I'm going to talk about is a model developed by my postdoc, Pierre Damien, who has been working with this coupled NEMO. I think everybody's aware of what NEMO means, this hydrodynamic recirculation model developed by at Europe, essentially, and then the biochemical model, the Pisces model, which is pelagic interaction scheme for carbon and ecosystem studies. I even remember that. So the configuration that we have is a model that has about a 7-kilometer horizontal resolution, 75 vertical levels, and then the biochemical model is a relatively sophisticated model, the Pisces model, with 24 compartments. It has phytoplankton. Phytoplankton growth is limited by 5 nutrients, and we have 4 living compartments, 2 for phytoplankton and 2 for zooplankton, and also the same for the detritus. And then, well, the model is forced by standard data, like at the open boundaries, we use this reanalysis that is called GLORIS, produced by Mercator, the Mercator group, and then we use the ERA interim flow, wind surfaces and heat fluxes, and then we use the World Ocean Atlas for the climatology from the World Ocean Atlas to specify open boundary conditions for the biochemical model. And so, just to show you that the model sort of, I cannot show you everything, but on the physical part, you see at the top the kinetic energy at the surface produced by Aviso, the sea level data from Aviso, and the geostrophic velocities from Aviso, and then below what the model does, and essentially, we have several physical tests to check that the model is doing a relatively good job. And then what's new is what we do with the biochemical model. So here is a vertical profile, vertical profile of nitrate and phosphorus, or phosphate, sorry, and then what you see in red is what the model produces. The dots are some profiles from this program that SIGOP, all these stations that we are carrying out in this project. And the other one is the climatology from the World Ocean Atlas. And the model is not perfect, as you can see, and it has some drawbacks. But we claim that essentially we are within the the observations, and that essentially the profile, the vertical profile, is producing or doing a relatively good job. We have big discussions on this with other modeling groups here in Ensenada who think that this is not as good as we claim. But let me leave that apart. This is comparing the seasonal profiles of chlorophyll with depth from between the model and cargo floats, and again, from the left to right, spring, summer, fall, and winter. And I have, well, again, just, I can only claim that the model is doing a relatively good job, is within the observation spread of the observations. Again, well, the maxima is not really reproduced, and the depth also is not at the right place. But we say that the model, or we think the model is producing or reproducing essentially the variability in a proper way. From that came another paper which actually put some finer things on these results that I showed you before regarding these integrated chlorophyll content and the surface content. So again, here on the right-hand side, you see these pictures for summer and winter. On the left, you see the model results. On the right, you see the satellite data. And again, you see that the modeling is doing a relatively good job in summer, and winter it actually overestimates the chlorophyll content at surface. The figure at the top is surface chlorophyll content at the surface time series, and you see in blue is the model, in red are the observations, and you see again that in winter the model is overestimating surface chlorophyll concentration. But what we found from the analysis of the model is that this picture of a system in the Gulf or the Gulf of Mexico as a broad system where integrated chlorophyll can change during the year. Well, in the model, actually, as I said, put some finer things here, and essentially we came up with a regionalization of the Gulf of Mexico based on the behavior of the mixed layer depth and the behavior of the integrated chlorophyll content and the surface chlorophyll content. At the top, you see integrated. On the left-hand side, you see the integrated chlorophyll content during the winter, and on the right-hand side, the integrated chlorophyll content for summer produced by the model. So essentially, what you see is that, or we came up with this regionalization in which you have the region 1, which is it's part of the Caribbean and part of the southern Gulf of Mexico, where we don't actually see an increase in the integrated biomass, although we do see an increase of surface chlorophyll concentration at the surface during winter. In the second region, which is the one in the middle, it's where all the loop current eddies actually travel along the Gulf of Mexico. And here we have a mixed kind of result, where I will show you in a moment, in which depending on what kind of eddies are found in this area, you will see an increase in the integrated chlorophyll content, or you won't see that. So it's a mixture of results regarding that. And then the blue one, which is the northern part of the Gulf of Mexico, where you always see an increase in biomass during winter. And well, that's more or less the results here. And this is shown in the time series here. So Here, this is the climatology again produced by the model. At the top, you see surface chlorophyll concentration. At the bottom, you see the integrated chlorophyll concentration. So what I was saying is that in the in this yellow region, you don't have an increase. The integrated chlorophyll is relatively constant, although you do see an increase at the surface. Then in the middle region, you see that okay, there is an increase in both, but this has to be quantified in a moment again. And then the blue region, where you do see a clear increase both in surface chlorophyll concentration and integrated chlorophyll concentration. And what happens in this middle region, which, as I said, is a region where these large anticyclonic eddies and a lot of cyclonic eddies associated with it are found most of the time, is that the previous, this figure again is a time series produced at model 1 point. It is region 2 or middle region of the Gulf of Mexico. The colors are the chlorophyll concentration, the red line is the mixed layer depth, and the black lines are the isopyctals. So what you see is that this is dominated, or this time series dominated by certain events, which are marked with the arrows there. And those arrows mark events in which these big anticyclones from the loop current were crossing this point where we're looking at this time series. So what happens in this middle region is that depending on whether you have one eddy, one big cyclonic eddy or not, will have a different response in terms of the integrated chlorophyll content or the surface content. And so essentially what we are saying is, well, this seasonal variability that is found in the surface chlorophyll content and the integrated chlorophyll content is really controlled by the mesoscale. And so we have really to look more into what happens inside of these eddies. And something that we presented in a meeting a few months ago here in Puerto Vallarta, which actually everybody is invited to participate next year, is the Mexican Geophysical Union. We actually, Pierre came up with some results which are apparently paradoxical in some sense regarding the usual picture that we have considering the anticyclones as regions of low productivity. So one of the things that we did is, in these pictures from surface chlorophyll, you can actually see very clearly all these blue regions, which are the anticyclonic regions of, or within the Gulf, and really mark the presence of, here the loop current, which is this area here, and then the anticyclonic eddy that separated before from the loop current. One of the things that Pierre did was to actually try to track both anticyclonic eddies and cyclonic eddies, and then do a composite of all of these, and then look at what were the conditions, in the geochemical conditions inside this eddy. It's the first thing is, again, that you will see in a moment, is what we have been, or what I've been showing you before, is that you really have to be very careful in trying to think, or thinking that what you see at the surface is what happens below. We find that is not really the case. So, first in this tracking mechanism, and then doing the composites of all the cyclonic eddies, which are marked here by CE in red, and the anticyclonic eddies in blue, is that, first of all, what you see is what I was saying. You know, if you look here, this is, again, this is chlorophyll at the surface. This is integrated chlorophyll concentration. But you see that these cyclones have slightly more chlorophyll at the surface than the anticyclones. But look what happened. This is winter. But look what happens if you look at the integrated content. I mean, what you find, and we claim that this is what we saw in the observations, and this is what we see now in the model, that the integrated chlorophyll concentration is actually higher in the anticyclones than in the cyclones. So this is kind of interesting. You know, this is against some way the common perception of what happens in anticyclones. And you know, to see what's going on here, did kind of also or look at the composite of the azimuthal or orbital velocity profile within all of these eddies. And so he came up with a differentiation also in kind of 3 regions. The first region inside all these anticyclones is a region where you have essentially solid body rotation, which is marked in blue. So this is, you know, well, what I just said, just like the inner core, these anticyclones are rotating as a solid body. Then the middle region, which is where the this azimuthal velocity reaches its maximum and then decays, and then the external part of the eddy, where really the influence of the eddy is really you know, fading, if you like. And this is mentioned here. So this the radius of the inner core is about 50 kilometers. The exterior region is, you know, larger or longer than 200 kilometers, and in between 50 kilometers and 200 kilometers, you have this intermediate region. And so Pierre started looking again at the, you know, doing a composite of those anticyclones. He started looking at the surface chlorophyll, sorry, at the total chlorophyll concentration. And so what you see here in these pictures is, you know, the composite in all these anticyclones that he found in the model. I cannot find my mouse. So this happens somewhere. So you see that in core you do have low integrated chlorophyll concentration, whereas in winter you have high integrated chlorophyll concentration. So, you know, let me remind you again. I mean, this picture in which we said, well, overall, you know, all these harbor floats, who knows what were they sampling? And at, although we had a relatively uniform distribution of observations during the year, well, in its broad sense, it really, you know, if the algo float is really not sampling one of these anticyclonic eddies, then it will see this idea, or it will see this uniform concentration of both, sorry, of the integrated chlorophyll, and a lot of variability or seasonal variability at the surface. But if you look into one of these anticyclones, then you do see that there is seasonal variability in the integrated chlorophyll concentration. And particularly, it seems that at least in this model, which has, you know, 7-8 kilometer resolution, what you see is that the integrated content is really mostly at the inner, in the center of the eddy. So what is going on? I mean, this is, we are trying to show that here, that so the left you see what happens with the primary production now in the model, which you can compute separately from the chlorophyll. And you do see an increase of primary production in the inner core of these anticyclones, and less clear, in the exterior part of the anticyclone. And also, what you see, I mean, if we are allowed to do this, or we have we can do that, these kind of calculations within the model. You start looking at nutrient distribution in the vertical, and you can also separate the primary production in new and regenerated primary production. So what you see on this left picture here, this is depth, this is nutrient concentration. So what you see is in the inner core an increase in nutrient concentration in the inner core, and less evidence, this is more blue, which means less in the exterior field. And then in the primary production, when you separate it between new and regenerated, you see that in the inner core, during the winter, the primary productivity or primary production is really dominated by new productivity or new primary production, whereas in the external part, this is not, it's not as evident as in the inner core. And the other important thing is during the summer, it's really the regenerated primary productivity that is dominating the variability there. So this is why you see that the red line, which is the rated primary productivity, is higher than the blue one. If you look at, why it's happening, well, Pierre came up with this explanation, which looking at the lifetime or the life cycle of one of these anticyclones, and then the top figure is the time series of the depth anisotropy. So the first part really represents the period when this big anticyclone in the current is forming, and so what you see is that the isopycnals are increasing in depth. And then once the eddy separates and begins to travel to the west of the Gulf of Mexico, well, it begins to decay. And so what happens is that the isopycnals start to, to decrease in depth. And so what we have is a contribution for explaining this inner core high productivity maximum, if you like, is that two mechanisms apparently are at work. One is, if you like, the eddy pumping mechanism associated with this anticyclone decaying. So an anticyclone decaying means that the isopycnals will start to decrease in depth to shallow, but also you have the effect of the wind in the anticyclones. And I don't know if you are familiar with this, but the idea is that if you consider the stress at the surface and compute actually the stress not only with the wind, with the wind stress, but subtracting the actual surface velocity and surface water velocity at the surface, which is the theoretical value of the stress, then what happens is that you have a divergence at least in the Gulf of Mexico, because of the dominant easterly winds, westerly winds, sorry, not easterly, but the trades. You have that the anticyclones are actually producing divergence at the core of the loop current or of the anticyclones because of this effect of scattering or subtracting the surface velocity, the surface water velocity from the wind velocity to compute the actual stresses. So this is one of, or the current explanation of why we see this maximum at the core in the primary production, etc. The results, I think, I couldn't see Enrique's talk, but I think the results are consistent with what is found with the gliders too. And you see a picture here, here the one here is chlorophyll, and you see these events as well, and the UV chlorophyll maximum. And what Enrique found was that there was seasonal variability inside the anticyclones that he sampled, or the group sampled with the gliders. So we kind of obtain a similar kind of result, or a similar result with the models. The only thing that is different is that he also finds, a lot of variability and important vertical velocities, if you like, at the at the rim of this piece. And what we say, well, this has to do, and we to look at this kind of variability, which is more frontal. So this scale variability has to do, or needs to be done with a higher resolution model. So this is what we're doing now, trying to look at the higher resolution physical model. So instead of 8 kilometers, now we have a model which is about 2.5 kilometers, and we are looking into this right now. What are the differences? Let me show you similar. I mean, I won't go into all these pictures, but we have another model developed by Sheila Estrada, which is a ROMS model coupled with a less sophisticated biochemical model. It has only 7 variables, and it was developed by Katia Fennel. And she has been looking at different processes. One is the upwelling in this Yucatan area with this model, and looking at the reasons why we see this upwelling and this introduction of nutrients into the near coastal zone, and the whole coastal area, if you like, marked there in black, which occurs particularly in spring, winter and spring, and we claim, or she claims, this is related with the behavior of the loop current. She has also been doing these simulations with high and low resolution. Low resolution is on the left-hand side of all these pictures. High resolution is on the right-hand side, and you see, interesting things that, for example, the mean kinetic energy is not that different between models, but obviously, for example, because of the high resolution, the vertical velocities are really very different in the high resolution model than in the low resolution model. Mixed layer depths are actually also not that different. But anyway, just to finish, let me just say that we're currently looking at or doing comparisons between the high resolution modeling results, also looking at sensitivity to to some different wind products are important here. And we also have a new Loris analysis, which is supposed to be a lot better than the one used before. So this is what the things that we are doing. Pierre has been working with a couple of colleagues in France with using neural networks to determine nutrient profiles from physical variables and oxygen data, which it's kind of cool to see. I mean, from a physical oceanography perspective, because this nutrient data is really is scarce. It's difficult to obtain. And what we're looking for in this project that we're in right now is, can we use this ADCP, zooplankton vertical migration data in these models to look at them, or constrain them in some extra way? Can it be done? Is it possible? Is it useful? So thank you very much.
Thank you. Thank you.
Other questions? Any question? Okay. And we will go forward because we are a little bit late. It is good for
Okay.
you. And I will try. You have to stop sharing your video, and now I have to get. I will ask Pepe to share his video.
Okay. Do you still see my?
No, now I can see myself. Now I can see you, but I will ask
Pepe.
Pepe. Are you there? Pepe.
Like it's not there.
He's supposed to be connected.
No, solamente los
Very
No. Pepe?
Do you hear me?
Yes.
Yeah.
Okay.
Okay. Here I am.
Can you share your video? Yeah.
I saw.
And
Do you see it? You don't see it, do you? Okay. Well, you see it. Okay. Now you do.
perfect, perfect.
Yes.
Yes. Yes.
Io non posso fare niente.
Okay. Here it goes.
Can you please?
Is that okay?
Yes. There is a delay in the voice from my side.
Yeah. Well, the voice is gonna be delayed.
No, your
But do you
voice.
see the title and everything?
Wait. Can you please move the little window up?
Yeah, that one.
Let's see.
No, this is my one.
But this screen, remove the screen where you are.
Okay. Can you hear me or is not too loud?
We can hear very well, but can you move, please? Where my window where I am away.
Is this better?
Ahora español.
Is this better?
Yo era español.
Pero es que te la meta.
Not really.
Róbate.
Io non posso chiudere il suo.
Okay, maybe I just keep going. The presentation is very simple and I can do it really fast.
No.
Pepe,
Yeah,
what did happen? Vemos la pantalla con el video de aquí. A ver si puedes
Okay.
eliminar el video. Y ahora ya te escuchamos perfectamente.
bien pues. Ahí
Adelante.
va.
Ahí va.
Vámonos. Okay, this is a very simple presentation. It's just about the timing of these peaks. These peaks are what is the mean cycle in the vertical velocity due to the zooplankton motion in the water. So these means has a requisite that you have to align them. So if you have every cycle like in this figure, right, then you see actually the modulation of the day, the length of the day during the year. But if you align them given sunrise and sunset, then you are okay to make the mean cycle. So the previous results are on the mean cycle, so it's a function of hours of the day, but just between 1,000 meters and 1,200 meters. This is a result that already shown in Haren and Compton in 2013. And the three references for this work will be this report for Bohem, which has plenty of figures, the paper of Van Haren and Compton, and this presentation. So the previous data shows the those times, the time where the maximum velocity, vertical velocity occurs, either for the downward migration or for the upward migration. But now I have some more data that go further down. So we have some ADCB measurements and they go this way. All this is in red because this is very obvious, and I'll show you why. So we can extend the computations that we did in 2013 or something from the surface down to 1,200 meters. So these are like the previous results that they just go overall most of the column up to 1,200 meters. And this is one of the references for that kind of data. But here I have more data in this location, which is the Yucatan Channel. And we have these measurements, which last like almost 6 years, at least in this area of 1,300 meters. The thing is that we need a lot of cycles like that, 6 years, 1,000 or more cycles to get a sensible mean. You'll see why. In the near surface, say from 300 meters to 340 or something, the cycles are very clear, very much ordered, regular, but not so in the deep area. So even though there's a mean cycle, it's really obscured by a lot more noisier levels. If you see that the same similar information, but just on one record, some level, so the daily cycles at 300 meters are much more clearly defined than at 1,300 meters. Similar thing happens with the echo amplitude, which shows some very clear signals in the near surface, but not so much in the deep levels. Even though the data is so noisy at 1,300 meters, that's 1,227, but 1,300 meters, the spectra shows exactly the peaks at the diurnal and harmonics of that frequency. You can actually see very clear at 1 day and half a day side, increasing the energy. And this is the mean. Now you can actually see that there's a bunch of like horizontal changes because the amount of data is different at different levels. So every change in amount of data has a slightly different mean. But here what I'm showing is 2 cycles. So this is 48 hours. And as said at the beginning, here we specify 6 a.m. as the sunrise and sunset at 18 hours. These are the 24 hours of 2 cycles, 48 hours, 24 up to here, or 24. I just repeat the cycle to make clear that the downward pulse is very sort of blurry, not so clear as the upward pulse. The upward pulse in the mean is quite clear, well defined, and then we can time when does that occur. So the problems with the downward pulse is that it's maybe just several pulses at the same during the same period are just too noisy. There's also a problem with the fact that it gives us a vertical velocity, upward vertical velocity in the mean. So that's a strange feature, but happens. So
why is it that there's a upward beam velocity is not clear. So here is the data of that mean. And here I'm just plotting the dots on where the maximum vertical velocity occurs in the downward phase and in the upward phase. And what is usually done, or what I usually do, is make a fit of dots in terms of a quadratic polynomial. And these are the results. The fit time, the time at which the maximum vertical velocity occurs in the mean cycle. So there's only three conclusions in this work. First, neural cells extend the 0 to 1200 meters published data up to the upward pulse. For the upward pulse, at 14 and 50, the extrapolation of the upper layers didn't match well. Like you can see here, the extrapolation due to the 0 to 1200 meters, and here is the actual fit in the data from 1200 to 1450 meters. So there's a you can actually see this in the figure. In red is the extrapolation of the first 1200 meters. So there's a small correction to that. No? But this tells you when does the maximum vertical velocity occurs. And the signal in the daily cycle is very noisy compared with shallow. The normal pulse is either too wide or rather two pulses inhibited in scalar time. And that's what essentially what Van Haren and Compton show in another work. Let me just say that in the Pacific Ocean, the North Atlantic, and the Gulf of Mexico, the migrations are almost equally timed. That's all I have to say about this.
Thank you.
Do you mean that there is no difference in downward motion and upward motion? The timing is the same. Pepe, can you hear me?
Pepe, can you hear me?
I cannot hear you.
I cannot hear you.
You remove the microphone.
The signal is as noisy as the noisy signal in the echo.
I was just asking you if the downward motion is as the same timing as the upward motion.
I think you have to speak directly to the computer.
Can you hear me, Pepe?
Now I do.
Now, I was asking you if the downward motion has the same timing as the upward motion, in the sense that there is no quicker downward than upward.
Well, in the near the surface from near the surface to 1,000 meters, it's almost symmetrical, but not totally symmetrical. I mean, the advance against sunrise or sunset is different, the downward and the upward. It's not an up function exactly. But that gets all messed up further down, because downward motion is quite wide and irregular, and it's much clear the upward. The upward pulse shows 1400 meters very well, but not the downward pulse. Does that answer?
Yes, yes, thank you.
Any other question? No? Okay.
Next.
Okay. We will go to the next.
Thank you, Pepe.
Thank you, Pepe. And Helmut, are you there?
Yes, I'm here, but I'm still trying to figure out how to put my screen, desktop on your screen.
You have to. Sí. You have to say no. Have to go to Skype. Go. And now we are seeing your.
Here comes the expert. Wait a second. Here comes the expert.
Can you put the presentation?
Open the presentation.
Eso es que como que tienes el Skype en
En modo psicodélico.
Del Julio ahí con
No están diferentes. No están diferentes.
Ahora,
Okay.
perfect.
And Pepe, can you mute your microphone, please?
Yes.
¿Estás seguro? ¿Ya lo ven?
Sí, sí lo ven, ¿verdad?
Yes.
Sí.
Okay. That's wonderful. It's okay. I gave this title and I changed it a little bit when I saw that just about all participants in this workshop are physicists. So if you, I took the liberty to change a little bit the structure of the talk. Just confirm that you actually hear me.
Yes, very well.
Okay. So let me just make a few general comments from this, from the point of view of a biologist. And it might be relevant to what we heard before. So if you look at the distribution pattern of organisms, aquatic systems, obviously they're driven by physics, chemistry, but chemistry is not just nutrients, inorganic nutrients, it's also vitamins and pheromones and all this kind of things. And it's also important that you have other biological entities, like predators, for example. And the other thing is, what is different from physics is that the response of biological organisms is in many cases density dependent. So that's one thing. The other thing is that biology in general is dependent on the population history. Age plays a role. You know yourself that you don't grow after a certain age and you still consume and respire. And it goes obviously for all the different marine organisms. But for example, if we think of phytoplankton, phytoplankton has often aged. And order of magnitude, they go through 50 cycles of vegetative growth, and then they have to go through sexual reproduction. So suddenly you have a population that changes completely its physiological makeup. It doesn't grow, it has a different form, it behaves differently physically. So you have these complications that age. Even if you take bacteria have an age, and when they reach a few stage, they're more likely to have viral lysis, for example. Then another thing that you have obviously is periodicity and synchronization with physical cycles. That's not only daylight and moonlight, but you also have reports that they respond to tides, and then you have even annual cycles, and you have endogenous cycles, and these have to be synchronized with physical cues. So they don't respond conservatively just to the physical environment around them, but there is again this behavioral response that has to do with history. Then another thing, obviously, that is important are pathogens. So you cannot necessarily relate directly nutrient transport to chlorophyll, for example. You could do an experiment where you, for example, grow together phytoplankton and zooplankton at a very fast rate, and you have a relatively low concentration of chlorophyll, but a high production of zooplankton. So it's you cannot necessarily equate the biomass to rates in, for example, models. The models, obviously, they work more with functional groups, and functional groups, for example, in the model that Julio showed, he showed 2 functional groups of phytoplankton, 2 functional groups of zooplankton. Then part of these effects get sort of averaged out, but still it's something to consider. And then in ecology in general, the signs and magnitude of correlations between organism and environmental parameters can change because there's a state dependence of nonlinear systems. It's a very general thing. You can say you start with upwelling water that has nutrients, you have phytoplankton growing, and the zooplankton comes in, it reduces the phytoplankton, but it doesn't increase necessarily the nutrients, things like that. So there are many examples of that. So what I'm saying is that the distribution pattern of organisms in aquatic systems, obviously you want to model it numerically, but I think if you want to understand what is really happening, it is more complicated. Now, if that is obvious for organisms, it might not be that obvious for non-living organic particles. You have the obvious, I'm talking about just detritus and stuff like that. The driving forces, you have the sinking, that is more or less, can more or less be described, but it depends on density, which it's not that easy to define the density of the particles. But particles don't necessarily only sink, they can also float, because they can have gases, for example. They can accumulate, as you all have seen, in these surface lines that are produced by Langmuir cells, but they can also accumulate at density gradients as they sink and they reach the equal density, and they stay there. But it gets more complicated than that, because they can do a phase change. You computer surface polarity of hydrophobicity. And you know that in the adiabatic very well, because the mucilage that you find is produced from dissolved organics. So you have the transformation between dissolved phase and particle phase. And obviously that once you produce from dissolved phase organic particle, then it causes these secondary effects like agglutination of other particles and so on. And you have obviously passive transport and so on of the non-living particles as well. So basically, I'm just talking the next few minutes only about problems. Actually, this whole talk is about problems and interpreting data. And maybe I can show you how little we know as ecologists, really. So, for example, you all know about fish schools, no? And now you should ask your colleagues, well, what is it that really induces the fish to school? And you will get different answers. You get more efficient hydrodynamic propulsion, more efficient sexual reproduction, to counter predators. That's what you see here. You have a diver down here, and maybe it helps then the fish with the vigilance or to camouflage from electrosensory detection, which is something that I don't really understand, but it's in the literature. And then the argument is that it helps against predators in general. Basically, the idea is that predators have less of a chance to encounter fish if they're in groups. Then predators will go through a feast and famine process. But I have seen no models that show that schooling makes predation less probable, because on the other hand, the predator needs less energy to harvest, for example. So there's, I have no answers. I just am telling you that we have many questions in biology of very basic things that we don't understand, and that we cannot measure, for example, but that seem to be not easily measured, but that seem to be very important. For example, vitamins and pheromones, which are obviously very important for sexual reproduction in the water also, and the pheromones, not the vitamins. So we have that unknown. And now I'd like to mention a little bit about the deep chlorophyll maximum, because it's mentioned before. It's a common topic. And personally, I would argue that we really don't understand the formation of the deep chlorophyll maximum. Now, if you look at the literature, and I think a very good start would be Colin 2015, and you have these different, okay, let me start differently. You have a global pattern in deep chlorophyll maximum. Probably the best data on Morel, who showed the statistical relationship between the depth of the deep chlorophyll maximum, the amplitude of that peak, in different types of shore waters. The lower the peak is, the less strong it is, and typically in more oligotrophic waters, which is contrary to what we heard before this morning. Now, you can relate that to the light depth, and that's what you see here. And percent irradiance, basically you get the maximum at, which is not correctly shown here, the maximum at the 1% light depth. But for example, the argument that was mentioned is that that's where the nutrients come in, and that's why the phytoplankton grows there, and That's why it forms that maximum, because it still has light, but it has more nutrients. That doesn't really hold, because if you look at the photosynthesis, the specific photosynthesis per biomass, then that specific photosynthesis per biomass is not the one that light, well, it's much further up. So that argument doesn't really count. But then you have other arguments. You have the argument, for example, that at 1% light depth, there's photoactivation. That means that the phytoplankton makes more chlorophyll because it has less light, and it tries to compensate for that low light level. That also doesn't quite hold. Although Cullen shows it here, but I don't think, I'm not sure, these are theoretical data, so I'm not sure that would explain, or he doesn't even argue that really explains the chlorophyll maximum. So even if we would know, even if we, oh yeah, there's another argument for the chlorophyll maximum that I think is pretty convincing, is because the chlorophyll maximum is more or less at the nucleus then the question is if the phytoplankton that is sinking is not sort of stuck there at that depth, retained basically in their sinking and accumulating there. No? Okay, if you take that sort of very general observation that you have the deep chlorophyll maximum, the 1% light depth, where you find also the nucleocline and the pycnocline, then the question is, okay, let's assume that the chlorophyll max is stuck the nucleocline, which is also known as a pycnocline. Then why is at that level the 1% light depth? Because the 1% light depth is set by, it's basically set by the phytoplankton, by the phytoplankton above and the water column. So obviously there is some type of feedback that I think we don't understand. No? Okay, so yeah, so just to continue with this theme that we really don't know too much. Here you have the chlorophyll maximum, it's a pycnocline, the typical, and what you see is you have this orientation of phytoplankton in the horizontal. These are chains of diatoms, no? And that seems to be a typical, these are in water, in situ cameras that took those. So that seems to be a typical feature. You have this orientation. One explanation for that is that one possible explanation is that it helps the phytoplankton at that depth to intercept more light. Makes a lot of sense. We really don't know. But you see again, you have these structures at every size level, and one topic obviously is thin layers, that order of magnitude 1 meter thin layers. These are not random changes of fluorescence. These are structures, and there are even some old data that show that in different peaks there are different species here. So obviously you have a well stratified column because they don't get mixed up. You don't have this concept of a non-Lewis level that is moving those things up and down, and you have these very well defined structures. And in the end, you can even go to the microscales where you have the structures. The Azam picture is something that is very popular to show that you have even at the micrometer level. You have structures, and here you see, for example, you have diadem chain, in and around the diadem chain you have bacteria. So you have structure at every level, and obviously structures have an important functional role, but we don't, most cases, we don't understand that. Okay, but the topic, this is just to talk a little bit about problems. So we have vertical migration of plankton. Let me talk a little bit about that. Diving short-term for food. You have pot whales, turtles. Turtles can dive very deeply, for example, into the mesopelagic. And you have that as a vertical migration, just to talk about the range. But what is really the topic here is more the dive during light to escape predators by zooplankton. Maybe it's not just escape from predators, but another argument that has been made that because the water is colder, they reduce the metabolic rate at lower temperature and save basically energy that way. But you have exactly the opposite to what the zooplankton does. For example, dinoflagellates that dive in the night when you're getting nutrients, but they are at the surface during the day. And obviously the question is what controls that cycle. And Pepe has talked a little bit about these very deep cycles, and the obvious question is how could they respond to light at that depth? No? And obviously vertical migration is important because about the carbon pump, but that just to mention. So when we talk about this very deep vertical migration that Pep mentioned, we are not really talking about organisms, small organisms that are order of magnitude 0.1 millimeter big, that dive every day 1,000 meter deep or not, but we're probably talking chains of different populations that move down. And that would be the bucket brigade model. And the idea is that the surface population is controlled by light and dives to a certain depth and defecates, leaves organic material there that is then picked up during the night by populations that resides underneath. That's why it's a bucket brigade, because it adds sort of, it relays organic material from one population to the next. And what speaks for this model? Basically, the Pepe found the pulse of biomass migration, looking not at the speed of migration, but looking at the pulse of biomass moving, moves faster than the speed of the single particles that be registered by the ADCP. And so to explain then that you need staggered population where the population below the top one starts to migrate before that one reaches the depth of where it stays during something is wrong. During the no. Okay. Then the question is, well, how does this population here, for example, know when it has to migrate if it's not so light? And is it chemical cues, for example, that takes that sort of set the synchronization, or what is it? A little bit aside from this, to complement this model of the bucket brigade, there's this concept of microbial gardening. The zooplankton that is defecating, eating and defecating is, eating the defecated, not only ingests, but so breaks up, fragments the particles. And it has been proposed that this fragmentation then furthers bacterial growth. And then these lower populations here don't live necessarily just what is left over defecated by the population above, but is actually ingesting in large part these bacteria that are cultivated by the broken up detritus. Okay, that question, the bucket reality was a question of what synchronizes then the migration of this deeper population. And these are the these are data from Van Haar and Compton that Peppa already mentioned that show 1,400 meters. This diurnal pattern. This is echo strength and the solar time here, and these days. And the interesting thing is that, and I come back to that, and actually Pepper already mentioned that, is when you look, this is the echo strength, and this is velocity, migrating velocity, and what he notes, what they Van Haar and Koch note, is that there's a much clearer upward migration of the plankton confined in a shorter period compared to less clear downward migrations. Now, I'll come back to that and repeat basically what Pepper says, but the question then is that depth, there's very little light, 1,400 meters. What is really controlling the migration? And that's this enigma. And there are data from high latitude vertical migration at 71 degrees, 78 degrees, 89 degrees, and this is here, and you see vertical migration. This is echo strength again, and this is relatively shallow, 18 to 22 meters, and there seems to be a pattern in a diurnal pattern in the migration. At these altitudes, and more in the summer than in the winter, but it still persists. And at 78 degrees, there seems to be a stop then in December of vertical migration, but you would clear more easy. So that brings us to the light penetration with depth, or in general the light sensibility. And one argument that has been made, for example, is that at 600 meters, the noon light penetration in temperate areas is more or less equal to the bioluminescence background. And then you can argue, okay, that means that you have so much random bioluminescence background, and there's a Cherenkov radiation and so on. Well, how could they ever see a diurnal cycle of light with that noisy background? No, and but then Cohen, for example, argues that they respond to very low light, 10 to the minus 7 micromoles photon per square meter per second. We have in Sonata, we have order of magnitude 1,000 micromole photons per square meter second at the surface, no? And then there's a physiological argument. Well, obviously, the organisms need light sensors that are made out of single molecules that receives the photons and convert them to an electrical signal, you know. So I did a back of the envelope calculation, and you don't have to. You have to see this critical because I really did a very quick estimate. But let's assume that pigment, the rhodopsin, for example, typically has an optical diameter of 50 angstrom. So you can calculate. You assume that a photon that crosses that pigment is being absorbed and does some charge separation or in the rhodopsin, that these pigments don't have an antenna configuration where they collect things, no? And that you have 100% quantum efficiency. And let's assume that you have 1,000 micromoles per square meter second in PAR in the visible region at the surface, and then you have an attenuation of, let's say, factor 0.1 per 100 meters, no? So at 1,000 meters, you have order of magnitude 6 to the 10 to the minus 12 photons per square meter per second. Now, Bowen-Set is 10 to the minus 10 to the 10 photons per square meter per second would be the limit. If he takes the absorption cross-section of the rhodopsin, 2.5, 10 to the minus 17 meters square, you get at a rate of photon absorption of this molecule of 10 to the minus 4s per second. That means that molecule only receives order of magnitude 0.5 photons per hour. And they obviously you say, okay, well, it's very difficult to imagine that that you get a recognizable pattern of that some neurons can process to recognize a diurnal cycle with that low flux. So I just leave you with that question. I have no answer to that. I haven't seen that calculation, but more or less I think it's all right. And that suggests to me that obviously the light, there is finite light at the 1,000 meter depth, but I don't think it can serve to signal to zooplankton the diurnal cycle. Well, I don't know much about acoustic detection of organisms, but there's a clear concept of what is important. It's there, what kind of material the organism is made from, that has any mineral parts in it, or it has gas bladders or whatever. No? Anyway, it obviously it's related to the frequencies that we look at, that we use in the ADCP, for example, or the echo sounder to detect these organisms. What I wanted to say is that ADCPs are wonderful because they don't really interfere with, or echo sounders, because they don't really interfere with they're not getting close to the organisms, because organisms are very sensitive. And this is a towed remote vehicle, and you can see how this between 250 300, so it's 100 meters. you can see how the organisms perceive, in this case, this myctophied fish perceives this toad vehicle. And you can imagine you try to do, for example, net sampling, whatever, the difficult. I mean, this is well supported very well to get quantitative estimates of biomass and the structuring of vertical structuring of biomass. You have even at depth, you have structures. It's not if the bathypelagic has structures, the vertical structure. It's not the homogeneous, completely homogeneous kind of environment in terms of organism distribution. So that was just general comments, and now I get to the point there, the things that I wanted to talk about, but it's just very quick. I would like to talk about some particles and minimum zones. In this case, the red would be suboptimal waters at 700 meter depths. And the crosses is tuna fishing, and I would like to show some data from Peru in this region. And these are LADP data, and you get with LADP data because you don't have to do the correction for attenuation of the sound with distance. You get a higher vertical resolution, and you can see the blue is the echo amplitude at night. This would be at night, and in the orange is the octane. These are data from 3 days, every few hours of profiles. This is some of this all these profiles and of the oxygen, and you see that it's an oxygen minimum between less than 100 meters and about 500 meters. And in the case of the LADP data, it's only the nighttime data here. But then when you look at the daytime data, nighttime data here, black, and when you look at the daytime data, you see these red data here, and you see that obviously there's this peak here in the middle of the oxygen minimum zone, this peak of organisms that migrated down here. And then you have this thing down here, this really big peak below this would be the oxycline, the oxycline, right here at the edge. And one explanation for this peak would be, for example, that these are organisms that are just waiting for the organic material to sink through the oxygen minimum zone, and then happily here in this water that has oxygen again, they wait to receive that feeding from above. Here this is just the higher resolution, same thing. And here you have LADCP data, again, over the different days and the different, and you see the diurnal cycle here up here. At night, you have these are the less biomass. You have less biomass here because it's all on the surface, and at daytime you have migration down into this region by this population here. But what you have is also a persistent population that stays, and that population stays there because there is this tiny little peak of oxygen here. Now, if you look at the numbers, this is really very low oxygen. We are talking about order of magnitude 10 micromoles of oxygen. So they stay here and survive at that depth, that very low concentration of them. And then you have this huge concentration of huge biomass below 500 meters. In this case, 70% echo from the at this depth. Then I said, okay, here you have the same station again. You have this little oxygen maximum, order of magnitude, the maximum is at 10 micromoles. And these are now data from beam attenuation. And we see how the beam attenuation is situated below the, right below the oxygen maximum. And then when you look at the stratification here, you see how that corresponds, this peak here corresponds to higher stratification. It's a very low, obviously, there's this whole range of stratification. But in general, in this oxygen minimum here, you have lower stratification than below that, and sorry, higher stratification than that, and the white, I don't know if you can see the white here, that's the oxygen. You see that you have this sort of maximum here. This lines for 5 micromole, and then you have real, basically 0 oxygen in this region here and up here. No? So you have, it's the interesting thing is that you have this stratification where you don't really expect much stratification of, for example, beam attenuation, which might be that one explanation. It might be a bacteria growing, for example, nitrate fires that spire nitrate, or something like that, or it might be just an effluent layers that come into the shell, which has been reported more or less at the same depth, at the same place. Here you have this is beam attenuation. You get here's a maximum of beam attenuation. These are the oxygen values. Look at the very low range of oxygen values that you see here. Okay, and so that then you have the same effect with migration that you see with the LAPCP. We think that's probably zooplankton, and we do have even at big time you have persisters here. This population that stays here, and then at the daytime when you have migration down, you have vertical migration into the real oxygen minimum zone. Oops, sorry. Okay, so it's a little bit redundant. It's interesting to look at the relationship between the LADCP echo and oxygen, and you see that in the different parts of the minimum zone, very similar pattern, that actually these organisms that represent this this echo exist at very, very low oxygen concentrations. Remember that we have order of magnitude at surface, you'll have 250 micron per liter, and here we are at much less than 1%. No? The question is what controls that distribution, and it probably is physiological control, but I don't have clear data on that. But when we look at the this little octane, secondary octane maximum here. And these are the nighttime echoes from LSD. This is the oxygen here, the orange. Then you see this very similar pattern of distribution. And I sort of tend to think that that has to be some sort of physical control that relates to things rather than a physiological control, because you don't get linear physiological control normally. But I don't know. It's not a, I can't. I leave that here. Now I just want to repeat what basically was the point, the main point of Pepe. I didn't know Pepe was going to talk about, so as you excuse me. So from the same cruise of Peru, we have the data from the ADCP, the shipboard ADCP. And what you see here clearly is this asymmetry in the distribution pattern of the active strain. These are attenuation corrected attenuation echo strength data. And in this case, you have okay. This is where you have this intermediate population that persists here at the intermediate oxygen max secondary. But the clear asymmetry in the migration from, and this is the migration from below the oxygen zone to the top. And that obviously reminds of what our company reported for 1,400 meters. In this case, we're talking about 700 meters to the surface, much less. And these are different long-term statements, and we see that of Peru in different stations. And basically, I have no explanation, but I just wanted to underline what Pepe reported, what Van Harn reported, that we have this unexplained phenomena of this asymmetric migration pattern. And that's it. Thank you.
Thank you very much. Hello. Questions? Any questions, comments?
You put so many questions. Very good. Very interesting presentation, really.
Yes, because the physicists know it all, but the biologists know nothing.
Yeah, here is we have a mix of physical, chemistry, biology. So it was a very interesting talk. Maybe comment or something. We have also the modelers here. So
no, I think.
You know, I forgot to mention something important, and I didn't question that in talks before. I think you have to be very careful when you take chlorophyll fluorescence data and you try to integrate them, because and there I think corrections that you can apply, but you really have to consider that there's a very strong quenching of fluorescence near the surface. So basically, when you do integration over depth, you are underestimating the chlorophyll on the surface. And obviously, chlorophyll is equal to biomass, but even if you just take chlorophyll, you have to consider quenching of fluorescence near the surface, and you do get an underestimate of chlorophyll near the surface with fluorescence data.
If I can add, there is also
no problems.
no, but there is also the problem with the fluorescence that you measure also, you could measure not living, pigments, pigments that are not in living matter also, where you have the
Well, I think I would argue against that. No, I would argue against that. I don't think that extracellular chlorophyll or dead chlorophyll is really a very significant part of it. But the point is that you have basically it's a physiological response. The fluorescence. You have an antenna, and the antenna collects the light and sends it to the reaction center, and that fluoresces. And if the antenna is sort of disconnected from the reaction center, you get less chlorophyll, and that you find that in the non-photochemical chlorophyll quenching or things like that. It's a very simple effect where the system, since the organisms cannot change the amount of pigments very quickly, they use another system to adjust the amount of energy, light energy, that is being led to the system. They open the valve or close the valve, and that's basically what you measure with fluorescence is after the valve. So you get a physiological response.
But you measure also for pigments with fluorescence. That also is a degraded.
No, you don't.
Why?
You don't. No, you don't. Well, two comments. Pigment you only measure with extracted chlorophyll, but even with extracted chlorophyll, the measurement of pigments is largely an artifact. I think most people are not measuring pigments anymore because they would be important. And what has been always before measured, the acidification method, was largely an artifact.
I have a couple of comments. One is, there are some diseases like ourselves think that biology is a lot more complicated than physics.
Yes. For sure.
But on the other hand, one thing is that I agree with a lot of what Helmut said. All these quenching and the comments you made about the fluorescence thing, I think that's taken into account in the processing of the cargo flows. You should, if you haven't looked at this paper that published, because there is a lot of data processing to kind of get the right, to get the right thing. And finally, we fully agree that these points are really caricatures of reality. But on the other hand, I don't want to pick the depth of the deep chlorophyll maximum and part of its variability. So they may be caricatures, but apparently they get something right. So I agree that the story is really very, very complicated. But the philosophy we use is that, well, if you can reproduce things with relatively simple models, then you can dwell afterwards on what the other things are doing. We might be getting the right answer for the wrong reasons, but something in favor of models, they do seem to capture some of the things that we observe. That's all I want to say.
Okay.
Thank you.
Thank you. Any other comment?
Yep.
Okay. I think for you it's in the morning, but here we are getting very late, and
They will close.
they will close us in the institute. So we really thank you for connecting you. I sent you the email if you want to follow also in the delayed mode the workshop. I send you the link to the YouTube. You can follow because of the lack in time, so you can follow afterwards in delay mode. Or if you want to stay up and you cannot sleep, you can connect and follow.
Oh, yeah, we received them. Thank you very much.
Okay.
Thank you to you.
Thank you.
Yes,
Bye.
thank you, Mike.
Bye.
Okay.
Thank you.
Goodbye. Thank you. Goodbye.
Bye.
Bye.
Bye-bye.
Ok. Come dobbiamo farmi? Io posso recuperare il mio computer? Allora,
The bus is waiting for you downstairs, just in front of the door. Sì, sì, adesso in what time do we meet? Because it's later
I booked for the 8 o'clock, the reservation.
we can call to.
Yeah, for the 8:30. I mean, you can make 8:15. When they wait for you.
It's okay for you?
It's near the.
Yes, 10 minutes walking.
We will meet on the
Perché una
restaurant.
sì.
No, we'll meet in the hotel. I will come in Milan in the hall and we walk
Okay.
10 minutes, 15 minutes. Ah, this is
Fine. What time? How long time do you leave France? 11?
7. Yes.
30 minutes.
Quarter,
40
yes,
minutes,
quarter.
30 minutes.
Go down, less than 40 minutes.
Esatto.
E alle 8:15 nella hall dello. Arrivo io e lì andiamo camminando.
Ok.